All right, I'm Hunter Black of the Coagulation of Erythrocytes group. I have 3 other partners that were not able to make it today: Delaney Cheriker, Maddie Bessinger, and Christopher Otap. We are from the Palmetto Scholars Academy in North Charleston, South Carolina. Background information: erythrocytes, or red blood cells, are the vehicles in which oxygen and carbon dioxide travel in. The platelets contained in the blood sample will coagulate at a certain point in time. Certain substances can be tested to understand the time before coagulation is initiated. This will hopefully be different than on Earth under the same conditions. And by conditions, in an American Red Cross blood bank, it is usually kept at about from 1 to 6 degrees Fahrenheit, and at that point it will last about 42 days. The question, the hypothesis, the question: how long will erythrocytes take to coagulate in a microgravity environment? And the hypothesis being, if erythrocytes are left in a container in a microgravity environment, then the erythrocytes will last nearly 21 days. As I said, in an American Red Cross blood bank, it'll usually last about 42 days, but considering that we can't refrigerate anything on the ISS, it's shortened to about 21 days before coagulation completely takes over. Materials. We will need red blood cells to be able to test how they are affected in space. We will need the additive solution to stop the blood from coagulating. In the American Red Cross, to keep the blood from coagulating and reacting with the plastic, there's an additive solution added to the plastic that allows the blood to circulate and not be affected by the plastic surrounding it. The analytical equipment is necessary to analyze the red blood cells and how they are affected. We will need to test if the RBCs lose technetium-99m, chromium-51, and indium-111. We will be partnering with the American Red Cross to receive the blood and the additive solution. The Red Cross will also allow us to use their analytical equipment. Technetium-99m, chromium-51, and indium-111 are all certain products of the blood cells that show vitality. So having those will show how far or how long it took them to coagulate. A regular blood donor has volunteered to give blood to be used in this experiment. The ground truth experiment. Since our experiment was not selected for spaceflight, our experiment could no longer conform to the research proposal due to financial constraints. Analytical equipment from the American Red Cross, nor the screened blood from the American Red Cross are not available to us. However, using type O blood, we have loaded a type 1 FME, which means no separate compartments, just one end cap filled with the additive solution, with 2 milliliters of the additive solution and 8 milliliters of the type O blood. This type 1 FME is being kept at room temperature in a secure environment. Blood can be dyed with propydium iodide to check for cellular viability. We're using the propydium iodide as a different way to check the viability instead of testing for technetium-99m, chromium-51, and indium-111 because we did not have the resources. We hypothesized that the red blood cells would live longer on the ground than in microgravity. The average lifespan of a red blood cell is 120 days on Earth. That's while in a human body, as it's circulating throughout the human body. The possible hypothesis, if our product had been chosen to fly, is understanding the coagulation rate of erythrocytes in a microgravity environment will allow for new knowledge that could lead to longer space missions or explorations, example given Mars. Certain acknowledgments we have. We're going to thank the partners and sponsors around North Charleston, including SPAWAR, CASIS, the Medical University of South Carolina as partners, and as sponsors we have ISHP, CASIS, and Palmetto Scholars Academy board members and the Academy families.